later. Sorry? In the middle of class when I finish. Yeah, thank you. Okay. All right. Good morning, everybody. Can you hear me? Okay. So welcome to Nutrition and Metabolism class. Make sure you are in the right classroom. All right. This is the first day. Okay. So my name is Shirley Chen. I'm a professor in the department. This department. Maybe I can, introduce myself a little bit about my research background. Our group is kind of interested in the obesity and diabetes research, and we're particularly interested in adipose tissue. Adipose tissue we're going to talk about later in this class. It's not just passively stored energy, but adipose tissue is very actively involved in the whole body metabolism by secreting number of interesting hormones or peptides that can regulate whole body metabolism, right? And for office hours, in my office in the 139 in this building, well, not this building, right? This ABMS building, the Food Science Nutrition Building 139, right? If you have any questions and concerns, you can just stop by anytime, right? Or if you wanted to, make sure that I'm in the office, so I'm available, so you can also make an appointment by email. We have two teaching assistants, Derek, Lynn, and Shara. So if you guys have any question or related to the grading or any class material, you can also contact them, right? Okay, so class today we meet in this room. The next Thursday we probably will change the classroom to 15, right? I will let you know by email, all right? And time the same, right? And we meet at 10:40 until 12:35. This is our course website. Are you guys able to access the web? Okay, so we usually post a lot of materials, class notes and other stuff and the model side. Course description. So this course is basically help you to understand the carbohydrate, lipid, and protein metabolism and the metabolic disorders. And we particularly focusing on the main metabolic pathways of 3 macronutrients, carbohydrate, protein, and lipids, right? And also we going to talk about their interrelationship or their interregulation, right? 3 macronutrients, they not metabolized by their own, but they also interrelated. And main concept we also want to learn in this class is the anabolic. What is anabolic metabolism, catabolic metabolism, and also so-called amphibolic metabolism, right? And how carbohydrate, lipid, and protein metabolized in our body under the different physiological conditions. For example, we will talk a lot, in the fat condition or in the fasting condition, right? And we also wanted to understand how macronutrient metabolism regulates to fulfill energy need for maintaining the normal physiological functions. We also talk about what is wrong, in terms of metabolism of these macronutrients in the obesity or disease condition, for example, obesity and diabetes, right? So the course objective. So after completing this course, you are able to describe the main metabolic pathways and processes involved in the energy utilization for body maintenance and growth and performance, right? And you also are expected to identify the different pathways, right, involved in the carbohydrate, lipid, protein metabolism and their interregulation in different physiological conditions, like I said, in fat and fasting condition, right? And also you will be able to assess the metabolic consequences, right, of deficiency or excess of carbohydrate, lipid, proteins. So what is going wrong if we eat too much, fat, carbohydrate, protein. And also finally, you should be able to evaluate information that acquired in scientific journals. You'll be able to read, you make a judgment or assess when you read the scientific journals or the other popular magazine, and form an opinion based on the knowledge that you learned from this class, right? Okay, reading materials. You will need have a textbook, biochemical, physiological, and molecular aspect of human nutrition, and Haber's electric biochemistry, which I think we're already available, right, on the Moodle side. And also we probably will use some like PowerPoint slides, basically class notes, right? You were able to get the class notes, and sometimes we probably will post some journal articles, a website that are related to our class. All right, so this is a metabolism class, right? So what is a metabolism? Can anyone tell me? What is a metabolism? What is the definition of metabolism? No? Just give a try. Anybody? Yeah.
See how things are broken down and used in our body?
Yes, yes. So it's something that, particularly in this class, what we're going to learn is something in the food, right? And then can be break down in our body, and our body can take up the small molecules that break down from the larger molecule in the food and take into the body and utilize in the body, right? So this sort of the simple process, right, the metabolism. So, and the breakdown and absorb into the body and utilize in the body, this processes all can be accomplished by chemical reactions, right? So chemical reactions is really, it's directly related to metabolism, right? So we learn metabolism, we actually learn the chemical reactions, right? So what food components can be used as energy fuels by our body? Thinking about it, what kind of food components? Nutrients, I would say, in the food that our body can take it and utilize as energy fuels.
Amino acids and glucose.
Yes, amino acids, glucose, and lipids, yes. So there's the 3 macronutrients that we're going to learn, right, in this class. So these 3 major food components or nutrients are the energy fuels that our body can use. What about the vitamins and the minerals? Our body able to utilize as energy fuels? I mean, energy fuels mean that, the molecule that can be metabolized and to produce ATPs. Do you think of vitamins, minerals? No, right? No. So vitamin minerals cannot be directly utilized for ATP production, for energy, but are very important factors that can modulate, a lot of enzyme activities and the chemical reactions, right? Now the next question, are we eating constantly? Constantly means like, you keep eating. No, right? We only eat like 3 meals a day, right? But our body needs energy constantly, right? So the question here, how do our bodies fulfill energy needs during fasting when we don't eat? For example, you're sitting here, you're not eating anything, right? But your body still need energy, right? How can our body fulfill energies during fasting when we are not eating anything? Can anybody? Yes.
It gets stored and then it catabolism if we break it down.
Right, wonderful. Yes. So because our body be able to store energy when during our eating, right? When we eat food and we utilize some as energy, but we store a lot of energy from the food in the body. So during the fasting, when we don't eat, and we can break down the stored energy in the body and utilize food energy during the fasting, right? So store energy in the fat condition, and this process usually defined as the anabolic metabolism, right? And you store energy during the fasting to break down stored energy and utilize them, and this process usually considered as a catabolism, right? Okay, so this slide I think give you a little bit better view of the whole process. What's happening in terms of metabolism in the fat fat or fasting condition, right? So for example, in the food, you have a lot of carbohydrate, lipids, protein. These are the energy fuels, right? And this energy fuels usually present in the food in a very complex form. So our body cannot be easily to take it into the body, right? If we don't break them down into the smaller molecules. So the digestion and absorption happening in the gut system, right, when we eat food. And so this purpose of digestion absorption is to break down complex molecules of macronutrients into small molecules, for example, glucose, fatty acids, amino acids. And these are the smallest molecules that our body are able to absorb into the system, all right? So the breakdown, larger molecules to small molecules, is the digestion absorption and then uptake into the body. And in the fat condition, we break down some small molecules, utilize some small nutrient molecules for ATP production, right, to provide energy for body use. So catabolism can also occur in the fat condition, but the majority of the glucose, fatty acid, amino acids that we absorbed from the food are converted to the large molecules in the body as a stored energy, as a form of stored energy. For example, glucose can be made to the glycogen, right? And fatty acids can be formed triglyceride, and amino acids can be synthesized. Protein can be synthesized from amino acids. So this glycogen, triglyceride, and protein are the stored energy, right? All right. So in the body, and so this process you form large molecules from small molecules, and this process is called anabolism, right? And catabolism is the large molecules that break down to the small molecules. This process is called catabolism, right? So during the fasting, our body still needs energy, and because we have a stored energy in the body, right? Although we don't eat, we don't have nutrients from the food, but our body has the stored energy. So we break down glycogen, triglyceride, and protein into the small molecules, glucose, fatty acids, and amino acids. And these small molecules can be utilized for energy ATP production during the fasting, right? So is that clear? Kind of relationship between the catabolism and anabolism depend on the physiological condition under the fat or fasting condition, right? Okay, so how to learn metabolism? Metabolism is biological reactions or biochemical reactions, very complicated, right? And have you guys all completed biochemistry, right? So I guess probably pretty hard, right? Biochemistry, very lot of things you need to memorize and very complicated. So and for this class, I just want you guys to focusing on the big pictures and the small details, right? To it probably will help you to learn metabolism. So you probably wanted to change the way that you learn, you used to for learning other classes, right? And try to focusing on the big pictures and the small details. So what are the big pictures? So big pictures include the pathways and pathway relationships. For example, when we will learn later, for example, glucose can be metabolized through 4 or 5 different pathways, right? And so you just wanted to remember how glucose be metabolized through which metabolic pathways and under the different physiologic conditions. Let's just take example. In the fat condition, when we eat food, and glucose can be absorbed into the body, right? And one important thing is that glucose need to be break down and complete oxidized to CO2 water and produce the ATPs. And this process of metabolic pathway is called glycolysis, right? Glycolysis is one important the catabolic pathways of glucose. And the purpose of a glycolysis pathway is to produce ATPs for energy. And the second important pathway is glycogen synthesis, right? We also need to store energy in the body. So glucose can also be utilized for synthesis of glycogen. So this metabolic pathway is called glycogen synthesis pathway. So glucose can be metabolized through the different pathways depend on the physiologic conditions, right? And during the fasting condition, our body needs to utilize stored energies. We need to break down glycogen, right, into the glucose and then oxidize glucose for ATP production. So glycogen breakdown is also one of the glucose metabolism pathway, but this pathway occurs under the fasting condition, right? So you try to remember this, pathway. This pathway we consider the big pictures, right? So try to understand how nutrients are metabolized in different metabolic pathway under different physiologic conditions. And most important thing is that how glucose metabolized in different pathways regulated, right? We know that in the fat condition, glycogen can be synthesized from glucose, but very important hormones, for example, insulin. Insulin is a hormone that can stimulate glycogen synthesis. So under fat conditions, our body have a higher levels of insulin. So that's why the glucose mostly be pushed to the glycogen synthesis pathway, right? So in addition to the metabolic pathways, the hormonal regulation of the pathway activity is also considered the big pictures, right? So big pictures, the pathway relationships, and the hormonal regulation are considered big pictures. And what are the small details? And if you wanted to go a little bit further, the details, how glucose being oxidized through the glycolytic pathway. So glycolytic pathway have many biochemical reactions, right? And we wanted to just learn the key reactions in each pathway. And key reactions catalyzed by key enzymes, right? So the small details that you need to remember that key reactions in each metabolic pathways and key enzymes that regulate those key reactions, right? So you also wanted to remember, these small details, right? So, and you also wanted to go back when you learn small details, or wanted to go back to the big pictures to comprehensively understand, how nutrients metabolize and how nutrient metabolism is regulated in the body during fasting conditions. Right? So this kind of things that you will learn in this class, right? Any questions? Okay, so now let's talk about the course assignments. We have a regular course assignment. We have a quizzes and midterm and final exams, and also formal writing assignments, right? And in addition to regular course assignment, we have a writing assignment. But I wanted to make sure that you understand the writing assignment only for people that need to finish the writing intensive requirement. If you already finished or completed writing intensive requirement, you do not need to take writing assignment, right? You understand? How many of you actually need the writing intensive? Not many, right? Maybe 8 or 7, something. Okay, so only. I'm not sure. Have you guys also taking the community, the community nutrition, right? So community nutrition also is a writing intensive course. So, but if you're taking community nutrition, you probably don't need to take this writing assignment, right? So in general, for majority of you guys, you just need to finish the regular course assignment, right? Okay, let's see. Oh, so for informal writing assignment, this is everybody need to finish, all right? So informal writing assignment, I usually will give you, the objective of this assignment is to assess your own understanding of the course material. So usually I will give you maybe 15 or 20 minutes, at the end of the lecture, and then give you a comprehensive question that related to the material that I just, delivered, and you can discuss with your group members, and then you write something to try to answer the questions and turn it in, right? So you will need a paper and pen to finish this, right? And turn the assignment in before you leave the class, right? So it's an open book discussion. And we have 7 quizzes, in-class quizzes, right? Just the very general questions, maybe 3 or 5 questions, right? But you just need, you may drop one at the end of the, semester. And we have 4 exams, including 1 final, so 3 midterm exam, and 1 final exam. Question, yes? Oh, it's just short answer. Yeah, short answer questions, right? And exams, all exams are multiple choice and forced choose selections, right? And also we have 1 homework assignment. I will give you 2 homework assignment, but you only need to complete 1, right? So you can pick which one you will like. And this is also a group work. You work together with your partners and get it done. And you also turn in as a group, right? The 2 people work together, and you just need to turn in 1 assignment. Okay, so writing assignment I just mentioned only for people who need to complete the writing intensive requirement, right? You guys told me, oh, can you guys send me email if you need a writing requirement, all right? And so for this assignment, a research paper presentation, how to finish this research assignment will be given on the September 12th after class, maybe 10 minutes or so. Dr. Renee Rosen will come to the class and explain this assignment to you guys. But again, only for maybe 7 or 8 people, right? You can. All right, grading. So your grade for the course will be determined based on a total of 410 points, right? 3 midterm exam, 55 points each, for a total of 165 points. And final exam, 80 points. 6 quizzes, 15 points each, for a total of 75 points. And you may drop 1 quiz at the end, right? Remember, you were given 7 quizzes, right? And 6 informal writing assignment and 10 points each, for a total of 60 points. And homework assignment 30 points, right? And in terms of contesting the grade, you give us like 24 hours, right? Read your answer again and write down whatever, explanation for you, disagreement with grading. You can talk to the contact TA if you have, or me if you have any concern or question about your grade, right? And makeup exam and late papers only under the very serious circumstances, all right? The document would be appreciated if you could provide me a document so that you can come to the class to take exam. Tips on how to succeed. So I assume you guys all knows very well, right? Usually come to the class and sit toward the front of the room and take notes. And I think it's very important for this class because of the, complex nature. Form the study group to prepare for the classes and exam. So group discussion is really very important and really, promoted this process. So, and also try to read assigned materials and review notes before class, right? Come class with questions and leave class, to make sure that you learn the key concepts and also thinking about some questions you probably still remain. All right, so and disability policies university, right? So if you have any concern that you need some help, and just let me know, right? All right, so the next thing that we're going to do is a background test. We have some questions for you. See if you're able to answer this question now or maybe later. So one thing I want you guys to do to form study group of 2, and this group will be the group that you guys work together for the homework assignment and a later discussion, during the informal writing activities. All right, and so you work together on this background test. And when you finish, and you can just write down the names, right, of your 2 people, and turn in the test as a group, right? So let's see what the time, 12, 11. Yeah, can you? So we can, we probably wanted to start back again at 11:50, right? So you just go ahead and work on the test, and then take a break, and then we start again at 11:50, all right? Sorry?
Now I'm like, I'm done now. Oh God.
One student asked to have a group of 3, but I don't know whether you think it's okay.
Well, I think it's probably 2. Just asking.
Just 2.
Oh,
Or do we have the odd number of the
I
students?
see, I see. Okay, that's fine. That's fine.
Okay. If you go on, so how much time you give them?
What's that?
How much time you give them to finish the background test?
Oh, I think it's okay. When we finish, we can just turn in, and we're going to start again 11:50.
11:50.
Yeah,
Oh, okay.
about half hour or so, I guess.
Okay, I see a list of questions.
Yeah. Yeah. I think it's enough time. I think so.
So we need to check all the background questions they answer.
I will take a brief look at to see how they answer. You don't need to do.
Oh, okay.
One per person or one per group?
One per group is fine.
Per group.
Yeah.
Oh.
Everybody got one per person at the back.
Oh, that's okay. That's okay. I made a copy so everyone gets.
I like it so far. Good class. Your organs are also in your stomach, though. I think they're all in that area. But, I mean, I do Yeah, that's what I mean. That romance, especially I think it's
Alright, so looks like most of you guys already finished, right? The test? So you probably don't need that much time. So I'm just thinking maybe you guys can take about 8 minutes break, then we come back 11:40, right? And also please turn in, to your test, right?
So everyone turn in or just one group turn in one sheet?
Yeah, just one group of one sheet.
Extra ones.
Oh, okay.
be happy that we have it. That's fine. I was going on it. Okay, I don't want to keep you involved in being. It's just she was funny. She was gonna just walk around with Kylie. So you get an A. I don't know. Because honestly, I am so I would okay, I'm I'm excited.
The work on is 3 different ways to Okay. Yeah.
It's regulated.
Oh,
Are we gonna go over this?
yeah, I can.
Okay. Maybe a group of 3 went up there is like an odd number or even number in the class. Instead of just
Yeah, we also have another big group of 3, so maybe you guys can
okay. Okay.
Yeah. Thank you.
You knew that though.
Yeah.
Okay. Thank you.
Hi.
My name is Brad. We talked a little bit at the beginning. I have a class that starts at 10:40 on Tuesdays as well. I'm doing like both classes at the same time.
Oh, yeah.
I was wondering if I could have one of those pieces of paper, the quizzes or the background test.
Oh, fantastic.
That would be great. Thank you.
These like graded. Oh, wait, this is not. Because then Wuhan is just regulated.
All right. Let's move on. So maybe we can go through this background test. I just took a brief look at your test. Looks like you guys did very nice job, able to answer a lot of questions already. So you guys probably don't need to go to class. Okay, so the first question is, does sugar turn into fat in what conditions? I think lots of you guys answer yes, right? So yes, sugar can be turned into fat under what condition? Fat or fasting? Fat condition, right? You eat sugar and you eat too much sugar, and then sugar can be converted to fat. So that's correct. And the second question, can your body synthesize glucose? Yes? Right? How? Yes, gluconeogenesis, right? During the fasting conditions, and we have a lower glucose level, so our body wanted to maintain blood glucose level and to make glucose from non-sugar precursors, right? Very good. So question 3, how does fat decide where to go on your body? Depend on, again, the physiological conditions, right? In the fat condition, we store fat, store triglyceride in the adipose tissue mostly, right? And also other tissue as well. And during the fasting condition, we break down triglyceride and move triglyceride from the fat tissue and break down triglyceride into fatty acids. And fatty acid can be released into the circulation and utilized by other tissues, right, during the fasting conditions. And number 4, where does your body store the energy? I saw a lot of your guys mentioned muscle fibers, muscle, right? And adipose tissue. Where else? What is the other tissue that can store energy? Very important tissue. Organ. Say loud. Liver, yes. Liver is a very important organ that stores glycogen. So liver, muscle, both tissues store glycogen, but the difference is we're going to talk about later. The liver is an organ that can also release glucose during the fasting. The muscle usually doesn't. Right? So liver, muscle, and adipose tissue are the 3 main tissues where energy can be stored, right? And number 5, what is the predominant storage form of fatty acids? Triglyceride, yes, triglyceride. And number 6, what is the predominant storage form of carbohydrate? Carbohydrate, sugar. Glycogen, yes, glycogen. 7, what does your body do to meet energy needs during a fast? Utilize stored energy, right? Break down glycogen, release glucose, and break down triglycerides, release fatty acids, and also break down protein and release amino acids, right? And what is the role of insulin? Have multiple roles, right? So in general, insulin is the anabolic hormones that can promote synthesis of a stored energy. For example, can stimulate glycogen synthesis, stimulate triglyceride synthesis, and also stimulate protein synthesis, right? So in general, it's the anabolic hormones and stimulate anabolic metabolism. And number 9, what would you expect insulin levels to be in fat or fasting conditions? In fat condition, what do you expect insulin levels? Will be high or low? High, right? Because in fat condition, anabolic metabolism is promoted, right? We want to store energy, so insulin levels high. And during the fasting condition, insulin levels be lower, right? And how is insulin levels regulated? Why insulin levels is high in the fat condition and lower in the fasting condition? What is the major or determining factors that regulates insulin levels? Do you guys know? Yes. Blood glucose levels, right, is a major driver or stimulating factors for insulin secretion from the pancreas, right, beta cells. All right, so that's all background test. So now let's talk about digestion and absorption of macronutrients. So in this session, you guys wanted to learn something you guys need to know. For example, which enzymes are involved in digestion. We know digestion process is to break down the complex macronutrients in the food, right, into the small molecules. And this process is enzymatic reaction, so needs enzyme to break down complex nutrients. And which enzyme that involve the digestion, and where are the enzyme produced secreted, and which tissues and organs secreted the enzymes, right? We know that very important places that produce these digestive enzymes, the pancreas, right, stomach, and small intestine. And where in the GI tract does digestion occur? And this kind of question you need to know. And what are the starting nutrients and components, intermediates, and the final products of digestion? And how and where are the final products of digestion absorbed? And how are they transported and to where? Right? So try to understand all these questions when you learn this from this session. Okay, so enterocyte, before we talk about digestion absorption, I wanted to point out that enterocyte is very important cells in the small intestine. That is a place where nutrients absorbed, right? So enterocytes has a unique structure. It's very different from the other type of cells, right? And mainly because of it has a brush bottle. So brush bottle basically is a microvilli, So and this structure is very important because it increases the cell surface, right? The surface of the cells, enterocyte cells, a larger increase the cell surface. So this is very important. That can increase the area that is directly in contact with the substrate, right, for absorption. So that can significantly increase the efficiency of the absorption. So enterocytes have a brush bottle, which is very important. And also I wanted to mention that on top of the brush bottle, there is called unsterile water layer exists, and this unsterile water layer is something that can prevent lipids to penetrate the brush bottle membrane and to the and then to the enterocyte, right? So because of this unsterile water layer, so the lipid absorption requires some special system to be absorbed that we're going to talk about later, right? Just mentioned here. And then also important structure in the bottom of the enterocyte is called basolateral cell membrane, and it's close to the capillary. So the nutrient that absorbed from the intestine lumen can be directly transported into the bloodstream, right, eventually to the portal vein. Okay, so now let's talk about the carbohydrate digestion absorption first. We have 3 macronutrients, right? So first of all, you need to know what kind of form of a carbohydrate in the food. In which form of a carbohydrate are present in the food. Monosaccharides, right? The food has a glucose, fructose, and galactose has already been, the small molecules. So these small molecules can be directly absorbed. They don't need further digestion. And second is disaccharides. For example, sucrose, lactose, maltose, and glucose. And this type of a disaccharides can be absorbed into enterocytes but need to be further breakdown to the glucose or fructose or galactose, right? And in the food, it also there are also the oligosaccharides. Oligosaccharides, some oligosaccharides are naturally present in the food, and other type of oligosaccharides can be produced during food processing, right? For example, maltodextrin usually is produced during the food process. And polysaccharides. Polysaccharides, for example, the starch, right, is a form of carbohydrate stored in the plant. The starch is most complex carbohydrate in the food. Right? This polysaccharides need to be break down into the small molecules. And lastly, also have some sugar alcohols like sorbitol and mannitol. Right? So I want you to concentrate on the polysaccharides because this group of sugars in the food need to be digested, right? Break down to small molecules and be able that can be able to absorb it into the body. So the starch. You can see we actually have 2 different types of starch, linear chain starch and branching starch. So linear chain starch, glucose unit, basically connected or linked by glycosidic bond in the form of alpha 1 and 2 linkage, right? So linear starch, also called amylose. And we also have another type of starch, branching starch, also called amylopectin. And the difference is that at the branching point, the glucose units are connected in the form of alpha 1,6 linkage, right? So digestion. So where does digestion of carbohydrate occur? Mostly in the duodenum and some in the mouth, right? And in terms of enzymes, what enzymes are involved in digestion of carbohydrate? First, what enzyme digests starch? Do you guys have any idea? Amylase. Okay, everybody knows, right? Amylase. So amylase is enzyme that digests starch. And amylase is produced by the salivary glands and the pancreas, right? So pancreas and salivary glands can produce amylase. Okay, so what enzymes digest oligosaccharides and the disaccharides? I can tell you this enzyme is produced by the anthracite. So amylase, alpha amylase, right, digests starch, and the alpha glucosidase, right? This is a name for a collection of multiple enzymes. So here are the group of enzymes that are collectively called the glucosidases, right? So all these enzymes are produced by anthracites, can digest the oligosaccharides and the disaccharides. All right. Any questions? All right. So now let's talk about the starch, the linear starch, amylose. How amylose is digested by amylase. So alpha amylase can break down the linear starch into the two products. One is maltose and maltose. Maltose is the 2-unit disaccharides, right? And maltose is 3-unit trisaccharides. And these two products can be further break down by another group of enzymes that we're going to talk about in the next slide, right? Eventually to the glucose. So amylopectin, which is the starch, the branch is starch. First of all, digested by alpha amylase, which is similar to the digestion of amylose, and to break down the amylopectin into the 3 products: maltose, maltose, and alpha-dextrin. Why the product of amylopectin has the alpha-dextrin? Why alpha-dextrin is produced in the digestion of amylopectin? Who is talking again? Okay, yeah.
Because it has the one alpha, one
Right, correct. Yeah, thank you. So because of alpha 1,6 linkage cannot break down by alpha amylase, right? So dextrin, see, you have still have a alpha 1,6 rich remain. So then alpha dextrin can be further digested by alpha dextrinase to glucose and maltose and maltose, right? So maltose can be further break down by maltase and glucoamylase, eventually to glucose. So the final product of this starch digestion is a glucose, and the enzyme involved includes amylase, right, and alpha dextrinase and maltase and glucoamylase, right? And depend on, of course, whether it's a linear starch or branching starch. Okay, so what are the final products of digestion? We already mentioned that, right? Glucose, fructose, and galactose, right? These are the final products of the carbohydrate digestion. Now the next question, how final digestion products are absorbed? Need to transport final product from the intestinal lumen into the enterocyte. Enterocytes, right? Enterocytes we mentioned is very important cells that does absorption. And this process requires the transporters, the membrane carrier protein, or called transporters, glucose transporters, to transport glucose from intestinal lumen into the enterocytes and eventually to the circulation portal vein. All right, so this is a slide show you how this is the whole process, the transport of glucose, right? A final product from intestinal lumen to the bloodstream eventually. So this again is the enterocytes, which is a free around, right? So this is the brush bottom membrane and facing the lumen, intestinal lumen, and then this side is the basolateral membrane facing the capillary, right? So on the brush bottom membrane, there are actually two different types of glucose transporters. One is called sodium-dependent glucose transporter, SGLT1, and the other glucose transporter is glucose transporter 5. And these two different glucose transporters transport different type of sugars, right? For example, the SGLT1 usually responsible for transport of glucose and galactose, right? And glucose transporter 5 is responsible for transport of fructose, right? And SGLT1 is called sodium-dependent glucose transporter, means that the energy for this glucose transporter to work comes from the sodium gradient across the brush bottom membrane, right? So how does this sodium gradient is generated? How is the sodium gradient generated? Basically, this enterocyte has a sodium, potassium ATPase, and this ATPase can pump sodium out of the cells, right? So makes the concentration of sodium higher outside of cells and lower inside of cells. So then create a gradient, right? Concentration gradient. So this concentration gradient is the sources of energy for transport of glucose by SGLT1, right? So that's why this SGLT1 transporter called sodium-dependent transporters, right? Does that make sense? Okay. And then on the basolateral membrane, the transporter for glucose is the GLUT2. Glucose transporter 2 that are located on the membrane, the basolateral membrane. And this GLUT2 is responsible for the 3 type of sugars transported through the little battery membrane and to the portal vein, right? So you probably wanted to remember this, different sugar transporters and transport different sugars and located at different place, right? Locations in the enterocyte. Okay, now let's do something. See if you can remember this transport process. So first of all, transport of final digestion products, glucose, galactose, are carried into the enterocytes by a secondary active transporter. What type of transporters? Glucose, galactose is transported by which transporter? SGLT1, right? Transporters, yes. And fructose is transported by facilitated diffusion through GLUT5, right? GLUT5, yes. And glucose, galactose, and fructose are carried out of the enterocyte into the portal vein by protein carrier GLUT2. All right, so okay, so now let's go back to the questions that we talked about at the beginning. So which enzyme for carbohydrate digestion absorption, which enzymes are involved in digestion? Do you guys remember? We just talked about it. Amylase. And if starch is the branched starch, we need additional enzymes, right? Remember this dextrinase, yeah, and then maltase, right, and glucose, amylase, right? Where are they produced? Where are these enzymes produced? Pancreas, yeah, salivary glands, right? Amylose. Okay. Where in the GI tract does digestion occur? Sorry? Small intestine, right? Which part of small intestine? Duodenum, right? And then some part of, the digestion can start, carbohydrate can start be digested in the mouth, right? Because salivary glands also produce amylase, right? Okay, so what are the starting nutrients and components? Starting nutrient. Starch, right? Starch. And intermediate can be starch or the starting molecules is starch or polysaccharides. And intermediates can be oligosaccharides, disaccharides, and triglycerides, right? And final product of digestion, glucose, fructose, and galactose. Okay? So how and where are the final products digestion absorbed? Where? In which type of cells? Remember? They absorb the glucose. Enterocytes, right? And how are they transported and to where? What kind of protein is needed for transport of glucose? Glucose transporters, right? So we talk about 3 different glucose transporters. Group 5, SGLT1, and group 2. And do you guys remember where are these glucose transporter located? In enterocytes. 2 are located on the brush border membrane, right? What are they? Group 5 and SGLT1, right? And we also have another one, group 2, and located, basolateral membrane, right? And transported to where? To portal vein, right? Circulation. Okay. All right, so we finished the carbohydrate digestion absorption. Now protein digestion absorption. You probably all know protein has a very complex structure, right? Primary, secondary, and tertiary and quaternary structures, right? And for protein digestion, first of all, the complex protein need to be denatured, right? And eventually to the primary structure, which is polypeptide chain, then can be digested by digestive enzyme and break down to the amino acids, right? So just wanted to remind you that this is the polypeptide structure, the primary structure. The amino acids can be connected by a called a peptide bond, right? And polypeptide can have a 2 terminus, N terminus, and carboxyl terminus, right? C terminus. And lots of digestive enzyme can break down the peptide bonds and release the amino acids from the polypeptides. So what are the starting molecules? We just talked about it, right? Of the protein digestion. Polypeptides, right? Primary structure of polypeptides. And what are the final products of the digestion? We have 1, 2, 3, 4 brands. So you thinking about 4 final products can be generated from protein digestion. You know, what is the final product for polypeptide breakdown by digestive enzyme and generate amino acids, right? Of course. So free amino acids will be the one of the products, and sometimes maybe they're not completely digested and can generate it dipeptides, right? That means you have 2 amino acids connect together, and tripeptides, 3 amino acids connect together, and then oligopeptide, more than 3 amino acids are linked together, right? And oligopeptides are not able to be absorbed because it's too large, right? Enterocytes cannot absorb it, so they need to be further break down in the lumen by brush bottle. So brush bottle can membrane actually is produced by enterocytes, but secreted, on the blood bottle membrane. There are 2 enzymes called aminopeptidase and carboxylpeptidase. These 2 digest enzymes are produced by enterocytes, right, and present on the brush bottle membrane. So then these two enzymes can further break down oligopeptides in the lumen of intestine, and then produce the free fatty acids and di-tripeptide. So di and tripeptides are able to be transported into enterocytes, right? Okay, are that clear? Any questions about the protein digestion? So again, the starting molecule would be polypeptides, right? And the enzyme can break down the polypeptide into the oligopeptide, tripeptide, dipeptide, and free amino acids. And oligopeptide need to be further break down at the brush bottle membrane by aminopeptidase and carboxylpeptidase, and these two enzymes are produced by enterocytes, right? And oligopeptides can be break down and produce free amino acids, di or tripeptides, and this product can be transported into the enterocytes, right? Okay, so where does protein digestion occur? So it looks like it occurs in the multiple places, right? And first, the stomach. Second, the lumen of small intestine and brush bottle membrane, right? We just talked about it, right? Of enterocytes, and then even inside of enterocyte, right? So again, can occur in stomach, lumen of small intestine, brush bottle membrane of enterocytes, and inside of enterocytes. So, and you also probably wanted to note, probably notice that the different places, the substrate are different, or the starting molecules are different, right? So in the stomach, basically, is the whole polypeptide, right? Digested, can be digested in the stomach, and the lumen, small intestine lumen polypeptides also are the substrate, right, for digestion. And brush bottle membrane basically digests oligopeptides, and enterocytes digest di and tripeptides, right? And eventually the amino acids can be transported to enterocytes and portal-vein eventually, right? Okay, so what enzymes are involved in the protein digestion? Do you guys remember? And the stomach, which enzymes that can produced by chief cells in the stomach and can be activated by hydrogen chloride to form pepsinogen. So pepsinogen are the enzymes that produced by stomach, right? And pancreas is very important organ that secreted a group of enzymes collectively called zymogen, right? And have 4 different type of enzymes can secreted by pancreas, and this group of enzymes, zymogen, and plays a very important role in digestion of the protein, right, or peptides. So trypsinogen, polylactase, trypsinogen, and procarboxylpeptidase A and B, right? So if you see something in the end of the word like gen or pro in front of the word, this means that this enzyme is inactive, right? So trypsinogen or pro-carboxypeptidase, this group of enzyme or zymogen basically is inactive when they are secreted, right? So they are secreted in the inactive form by the pancreas. And in addition to stomach and pancreas, the small intestine is also able to secrete some enzymes that can digest the proteins. First of all, actually, is a cholecystokinin. Cholecystokinin is the hormone that is secreted by mucosal cells of the small intestine, and this hormones, the function of this hormone is to stimulate secretion of zymogen, right? And also the enteropeptidase can be is secreted by small intestine, can activate trypsinogen to trypsin, right? And trypsin is the important enzyme that can activate the other zymogen we talked about earlier, right? Because zymogen, when it's secreted, is in the inactive form, so it needs to be activated. So trypsin is the one that can activate zymogens. So again, the small intestine can do two things. One is to release the hormone called cholecystokinin. Cholecystokinin is a hormone secreted by small intestine to stimulate zymogen secretion. And the second thing that small intestine does is to secrete enteropeptidase, right? So enteropeptidase is secreted to activate trypsinogen to trypsin, and trypsin is the important enzyme that activates zymogen, right? So kind of sequential events that eventually activate all the digestive enzyme for protein digestion. And finally, enterocytes, we already mentioned, can also produce two important enzymes, aminopeptidase and carboxypeptidase, right? And can digest dipeptides and tripeptides inside of the enterocytes, right? So multiple, places that can produce digest enzyme and work together to digest protein. Any questions? I know it's a little bit complicated right here. So I think next two slides I'll give you a little bit more detailed explanation how protein is digested, what kind of enzymes that produced by pancreas, the small intestine, how they work together to digest proteins, right? So first of all, pancreas. We know pancreas is the main sources of zymogens, right? And zymogen, we have 4 different type of enzymes, collectively called zymogens. And these 4 different type of enzymes produce, at the first place, they are in inactive form, right? They are not active. And they secreted into the small intestine. And pancreas also can produce the bicarbonate, right? And so here's a question: why is zymogen secreted in the inactive form? Zymogen is secreted by pancreas, right? Why they are secreted in inactive form? Do you guys know? Thinking about it. So zymogen, this type of enzyme usually break down digestive proteins, right? And we don't want it to digest in our own tissues, right? Our own tissue, also made up of proteins, right? So then zymogen is secreted in inactive form is to prevent self-digestion, right? And once it's secreted into the small intestine, then we want them to be active because we want this enzyme to digest food, not our self tissues, right? So once it's secreted into the small intestine, we want to activate zymogens. So how zymogen is activated? In the small intestine. So I mentioned earlier that trypsin is the key enzyme in here that activates zymogens. So first of all, the entropeptidase, which secreted by small intestine, can activate trypsin. So can cleave the trypsinogen to get rid of some protective peptides, and then to activate the trypsin, right? So this is the first step in the small intestine. And then trypsin is is the key enzyme that activates zymogens, right? So trypsin activities requires the alkaline kind of environment. So bicarbonate that's secreted by pancreas can help raise the pH in the small intestine environment, right? So then can activate trypsin. So trypsin is not active in the acid environment conditions, so it requires to raise the pH. So bicarbonate, the role of bicarbonate is to increase the pH to allow trypsin to be active, right? So trypsin then can activate zymogens to convert its inactive form of zymogen to active form of zymogens, right? So then eventually zymogen will be active and then to digest the protein in the food, not our cell tissues, in the small intestine, right? Does that make sense? So again, try to remember this. What is the role of a trypsin? What is the role of a trypsin? Yes, to activate zymogens. And what is the role of the bicarbonate? Yes, to increase the pH, to activate the trypsin, right? So they all work together to activate the digest enzyme and to allow the digestion happen, right? All right, let's see. What is the next slide? Okay. So protein, once it's digested, also need to be transported, right? So protein also requires transport system. I don't think you need to remember details, but just wanted to know kind of big things, two different type or two different type of transport systems, right, for to transport free amino acids. So first type of transporters called sodium-dependent transporter systems. For example, iron coupled transporter and electrochemical potential generated from the ion gradient, whatever called energized transporter. So just try to explain what is this type of transporter. This type of transporter requires, again, similarly like we talk, in the glucose, right? The sodium-dependent glucose transporter. They also require the ion gradient, and so sodium gradient, concentration gradient also is required for the transport activities of this transport system. So there are actually 4 different types of transporter in this category. One is called system B, and this system B usually responsible for neutral amino acids transporter of neutral amino acids, right? And ASC transport system basically transport alanine, serine, threonine, cysteine, glutamine. Okay, so and the immuno transporter system is responsible for protein and amino acids, and last one is the XAG transport system is responsible for aspartate and glutamate. So again, there's 4 different transport system specifically transport a specific group of amino acids, right? So just remember, general this sodium-dependent transporters have 4 different transport systems that responsible for specific group of amino acids, right? And the second group of transporter called sodium independent transport system as the system L, YL, and FC system. Again, they also transport a different type of amino acids. Different transport system transport different type of amino acids. All right. Okay, let's see. Yeah, I think we almost there. So, and then the transporter di-chip peptides are also specific transporters called a PEP1 transporter, right? And responsible both for the transport of di and chip peptides. And once inside of an enterocyte, and again we already mentioned, like this di and chip peptides can be further hydrolyzed by a aminopeptidase and carboxypeptidase, right? These 2 enzymes and produce inside of enterocytes into the free amino acids. So free amino acids eventually can be transported into the portal vein, right? And just one more last thing is that not all the amino acids that absorb from the food be all transported into the portal vein. Some of the amino acids, for example, glutamine, glutamate, and aspartate, right? And these 3 amino acids can be used by enterocyte for energy, right? So some of these amino acids, they were not transported into portal vein, but utilized in the enterocytes for energy, right? So absorption is greatest also in the small intestinal duodenum, right? It's a place that is, for the absorption of a large number of amino acids. All right, so we just want to stop here for today. And just for those of you for the writing assignment, please don't forget to send me email, all right?